You should see my screen now, and with the title of the PowerPoint. Okay, today I will speak to you about and also other viruses, and how we applied LC-MS into speeding up those kind of analysis, and also getting more insight than what people normally does. All right. So my background, I've got a master's degree in pharmacology, a PhD in biochemistry from University of Sherbrooke, and I had a chance to do a PhD at the same time with a collaboration in Lille in France in life science, where I learned a lot of mass spectrometry. And during my PhD, I had the chance to study the role of enzymes in the activation of the virus glycoproteins, and also study the cytokine storm induced by bacterial and viral infections. That led to a couple of publications. And after my PhD, I started a company offering services by LC-MS, and I had the chance to actually use my knowledge around enzymology and virus activation and the mass spec field. So today's goal is to have a brief overview of the role of the host endoproteases in the viral infection. I know someone actually spoke about TMPRSS2. It's one of those enzymes that can be involved. I will have you a brief overview of important viral gain of function related to host endoproteases, how stress coronavirus exploit the system, and how we can actually study this relationship, and how we actually use LC-MS mass spectrometry to improve science in that area. The take-home message is that you have to remember that the host protein virus interaction is a tricky system to exploit, and using LC-MS is a good way to improve our regular essays regarding those kind of analysis. So how glycoprotein or viral glycoprotein works, they will attach the virus to the cell, and most of the time, if you see my pointer, okay, it should be there. You should be able to see my pointer. There will be a site, like a hinge there, that can be cleaved to allow the glycoprotein to activate. And how it works is, once it's cleaved, it will be able to, when it enters a cell, it will be able to activate fusion of the membrane of the virus with the endosome membrane and release the viral content inside the cell. And there's an important gain of function that is well known to be linked to increased infectivity. Many viruses have used that mechanism. It's introduction of a multibasic sequence in that loop over there. You have an example there. And it's known to be activated by a family of enzyme that's ubiquitously expressed, named proprotein convertases. There's actually 9 genes. 8 of them are able to cleave at this kind of site, and 7 of them are actually active enzyme. One of the 8 is not only have activity once in his life against himself. And furin is one of the most used as a model, but that's probably a mistake of thinking that furin itself is like all the others. There's a much more complex biology behind that. So how stress and the actual stress coronavirus exploit that system. So you have the spike protein, which is the surface glycoprotein, that needs to be cleaved. Actually, there's 2 activation sites, 2 cleavage sites on the stress spike protein. And there's also the nucleocapsid protein that also needs to be cleaved to fully release the content inside the cell. So you see the cycle there, and you see that TMPRSS is involved in the cleavage. Furin is involved in the cleavage, and I'm not quite, how could I say, I'm not quite comfortable of showing it that way, because what I know from my studies is mostly cleaved during exit, not much during entry. And you have cathepsin also involved, mostly known to be involved in the cleavage of the nucleocapsid. And you see that family of enzyme there, cathepsin, furin, and they call it trypsin, but it's actually TMPRSS enzyme family. And you can see there for the spike protein, and you see the coronavirus OC43, the SRAS coronavirus 2, which is the one actively spreading right now, and the SRAS. You see that you will have the addition of the sequence over there, and also in the second sequence, multi-basic residues that can be introduced. And you see there in that publication, they were calculating a furin cleavage score, which is, I know what algorithm they use is not quite accurate, but it's a way of looking at this. All right. So how do we study that kind of interaction? In the paper I just presented you, it's published a few months ago, they actually use indirect approach where they look at co-expression of the mRNA of the coronavirus and the mRNA of the proteins and make correlation based on that. There's also people doing indirect analysis for colocalization using immunofluorescence. There's also other indirect bioinformatics approach where you use cleavage prediction models that are not always accurate, and also 3D modeling where you will look at this hinge and see if it can enter the binding pocket of an enzyme. And there are direct approaches where you will do some Western blot in vivo and look at the cleavage. Which is not quite high throughput method, but it's probably a good method to look at the protein, how it's behaving in tissues and in cells. And there's direct cleavage peptide in vitro assays that are done. Most commonly, it's done using either fluorogenic peptide with a single amino acid at a side, and some people have developed FRET approach, where the cleavage will break down the FRET transfer. The problem with those approach is that most of the time you cannot, if we go back a bit, you cannot work with very long peptides, and that's a problem. This is from the same figure we had. You see that the binding pocket in every position of the amino acid is very important. So it's important to work with longer peptides. And if you see there, it's a 3D structure comparing SRAS and SRAS2, where there's a gain of function here in the hinge with a much longer hinge compared to SRAS1. And this very long peptide actually has some kind of structure inside the protein and is much different than what you see normally in the protein. So it's very important to work with either the protein or with a longer peptide to study that kind of interaction. If you work only with those fluorogenic substrate, it does not represent the reality. So how people does it with a full-length peptide? So the classic way that has been published is using HPLC UV with a very long gradient where you will have your full-length peptide and you will look at the reduction of that peptide. And you actually need to separate it from the fragment so you don't have that interference there. And the problem with that approach, it's totally not built for in vivo because you will have too much material there for in vivo assays. So what we've done, and let's look there, we have a 60-minute runtime. Maybe with modern UPLC, you can shorten that to 15 minutes, something like that. But you will still have to be able to separate the fragments to remove the interference. If we go to LCMS, we were able to reduce down that to 2.5 minutes, and probably could have been able to improve it further. And the other important thing is that we can actually identify the cleavage site using MSMS sequencing, and the approach can be transferred in vivo thereafter, where we'll look at the cleavage of a protein in vivo. So let's look a little bit of this at how we did that study. One important thing first was to normalize the different enzymes together. So what we did, we actually tested a bunch of enzymes against the cleavage of a couple of peptides from coronavirus. So different variants, different enzymes, and see how those enzymes can be important or not in the activation of those coronaviruses. And to be able to normalize this, sorry, there's a typo here, we need to work at two different points. One is the Keyham and one is the Vmax. Why those 2 points? So here we can see it very, very well. With one enzyme, we have incubated with 4 different peptides, and we can see that at Keyham is actually the affinity of the peptide with the enzyme. So we can see that different peptides have a different affinity for that enzyme in particular. But if you look at the Vmax, which is the capacity of the enzyme to cleave, the enzyme is actually capable of cleaving that peptide, whatever is its affinity. So that means that, for example, that enzyme, whatever is the coronavirus activation site, if there's a lot of viruses, it will be able to cleave. If there's not a lot of viruses, in that case, it will not much be able to cleave the virus. It will not be very important. So this is the kind of study we've done. So if we look at 3 different enzymes there, we have done 4 different enzymes. So we have enzyme 1, enzyme 2 and 3 that were behaving exactly the same, and enzyme 4 there. You see, for example, for enzyme 2 and 3, we have only one peptide with low capacity, but high capacity, but low affinity with that peptide. If you look at enzyme 4, which has for every peptide very low affinity, but high capacity. So that means that enzyme, for example, sorry about that. If you look at that enzyme, it will not be very important in a viral initiation. It will not be very important there at the beginning of the infection. But at the end, when there's a lot of virus there available, a lot of substrate available, it will be important. And if you look at the enzyme 1, it's only potent on 2 peptides there, but not actually able to bind and cleave the other peptides. So that means that it's a sequence specific action there. So the other important thing we were able to look at is that, yes, there's that introduction of that multibasic residues. And if we look at 2 different enzymes, and we look at those multibasic residues, you could see that for some enzymes, it's actually able to cleave at different position in those sites. So that means that in some case, the multibasic residues is very important to be used for one type of enzyme, but for another type of enzyme, it will be able to cleave at those different sites, whatever happens. So you could probably remove one of those lysines and it will be able to cleave there anyway. So that's some very important information that we can gain from that. And more importantly, we can actually transfer that in vivo using exactly the same assay, but we introduce N-terminal labeling. So what it will give at the end after trypsin digestion to reduce the protein into smaller peptides for analysis, that we will be able here, for example, to look at the N-terminal, but at the actuation site, you will have 2 different peptides actually, one that will be smaller with a label there, and the other one that will be the full length. So we can actually translate that into in vivo assays and continue to monitor how the enzymes for example, with siRNAs, can activate the virus. So the conclusions there, based on what we've done in the lab with the different enzymes, and sorry, I cannot name all those enzymes because those data are confidential right now. The coronavirus seems to have a backup strategy to exploit hosts, so it doesn't use a single enzymes, and some enzymes are probably more important. Like we learned this morning, TMPRSS2 is probably more important for the viral initiation, which is probably the most important thing in infection. So that's very important to target the right enzyme in that case. All enzyme does not necessarily exploit the same cleavage site, so the virus has some kind of backup strategies to survive. What we cannot tell right now based on what we've done is it important to actually cleave at one place or the other in vivo? So that's something that should be answered. LCM-SMS has helped us to improve the speed and the amount of information we gathered compared to conventional plate assays with very, very simple substrates. And we can transfer that in vivo. So yeah, that is it. If you have questions, and this is the company I've created after my PhD, and those studies have been done inside the company.
That was a great talk. Thank you.
So is there any questions?
I think you gave a very nice talk. Thank you. I love it. You know, it's getting first-hand insights about the complexity of regulation. And I mean, it's great. Thank you.
Yeah, I actually had a question by someone who said, well, why don't we target those enzymes? Why there's no one doing it right now? And I said, well, it's probably lack of data and the fact that it's very complex, but it needs to be tried.